Hi, everyone. Welcome to the Johns Hopkins seminar series on the cerebellum. Today, as our guest, we have Amanda Therrien. Amanda is originally from Canada. She receives her bachelor's in human kinetics from University of Ottawa, then a PhD in kinesiology from McMaster University. She completed her postdoc training with Amy Bastian here at the Johns Hopkins School of Medicine, Kennedy Krieger Institute. And then she received a faculty position at Thomas Jefferson University. She's now an assistant professor there, where she runs the sensorimotor learning lab at the Jefferson Moss Rehab Research Institute in Philadelphia. Her research goals have focused on trying to understand the deficits in cerebellar disease and trying to use some of the remaining components of the brain, for example, the learning system in the basal ganglia, to try to improve the functions that have been lost with cerebellar disease. I'm really looking forward to hearing about the latest research. Amanda, thank you so much.
Thank you so much for having me. This is a very nice honor to share some of the work that we've been doing here. So let me just get going here. Good. All right. Yeah, so obviously I'm speaking to a crowd that is quite familiar with cerebellar ataxia, but that is the condition that a lot of my research aims to understand. Of course, it results from damage to the cerebellum. Although any one cause of cerebellar ataxia can be quite rare, as a phenomenon and as a phenotype, it's actually not very rare. Many conditions, the cerebellum, it's one thing I sort of love about the cerebellum. It's an identify with, it's a very sensitive structure, and it's easily damaged in a lot of conditions. So a lot of my work and the work that I'll talk about today has focused on individuals who have one of a series of degenerative genetic conditions, excuse me, but the most common causes of cerebellar damage are actually associated with multiple sclerosis, and it is a common cerebellar malformation is a very common side effect of premature birth. Now, as I mentioned, ataxia is a phenotype, and clinically, what designates a pure cerebellar ataxia is that you get this condition of very poorly coordinated movement without any concomitant paresis or peripheral sensory loss. So to give you an idea of kind of what anyone who might not be familiar with what these sorts of movements look like, I'll show you some sort of bird's eye view of reaching trajectories. So if I asked most of you to reach out to a target in front of you and I plotted the trajectory, had you do a couple reaches, they'd look something like this. So they're pretty straight line, and they're sort of movement is well calibrated for the distance to the target. However, if I ask some patients to make the same movement, you can see that the trajectories look quite different. Going from a more mild example to a more severe patient example, you see this sort of characteristic pattern of ataxia, which is you get this sort of low frequency oscillation that results, sometimes clinically it's referred to as intention tremor. I wouldn't really call it a tremor per se, but point is you see that these sort of very highly variable, highly curved movement paths. Now, as many of you are probably familiar, there's no medication or sort of other kind of traditional medical treatment that is widely effective for treating symptoms of ataxia. And so this leaves rehab therapy, so things like physical therapy, occupational therapy, speech language therapy, as really the sort of frontline options in managing this disorder. But for a series of complicated reasons that I think we probably don't fully understand, our rehab interventions don't work that well for individuals with cerebellar damage. And so an overarching question of my work has been, how can we improve rehab interventions specifically for individuals with cerebellar ataxia? And so in doing this, I like to think about kind of what do we think the cerebellum, a healthy cerebellum, is doing, and so I use this sort of cartoon example here. So, we know it's very important for movement control, and if you think about making a movement, we know that our motor cortex kind of needs to send. It doesn't pop over; it goes down, but send a command down to our muscles, causing them to contract. But the sort of central problem of motor control is that our brain has no direct knowledge of anything outside of it. Like it doesn't know anything about how the movement was performed. It doesn't know anything about the context in which the movement is being performed. And so it needs some form of information, and sort of an obvious source of that, we think of things like feedback, sensory feedback from the periphery. But relying on this information is problematic because processing can be quite slow. And so one sort of popular hypothesis about the cerebellar function is that it serves as part of a network that generates kind of these internal predictions of sensory feedback of movement commands that kind of serve as rapid internal feedback signals that allow for better coordinated and rapidly executed movement. Now, of course, these predictions can't exist in a vacuum. We live in a world that is constantly changing, and so the cerebellum has also plays this important role in a learning mechanism that keeps those predictions up to date. And we sometimes refer to this as adaptation or sensory prediction error-based learning. I'm going to walk you through an example of how, there's many ways to test sensory prediction error-based learning in the lab. I'm just going to walk you through kind of one example that's used a lot. So we'll often have people make reaching movements to a target, except we don't let them see their hand directly. We show them a cursor, and we map that cursor's movement to the movement of their hand, in this case, their index fingertip. And we have them make a bunch of reaches so that they form this mapping between the movement of the cursor and their movement. But one way we can test adaptation is by introducing a perturbation suddenly. And so I'll show you an example of one type of perturbation called the visuomotor rotation, where now the movement of the cursor is rotated in a consistent way relative to the movement of the finger. And the way adaptation learning or sensory prediction error learning works is that this now introduces a sensory prediction error. So now the predicted visual consequences of the movement no longer match what actually was the consequence of the movement. And we think the cerebellum can kind of use the vector information, so the direction and magnitude of this error, to figure out how to move, how to update your movement to account for the new sensory consequences. And of course, the way we test whether this learning has persisted or whether this actual recalibration has occurred is that we'll suddenly remove the perturbation, and we should see behavior that counteracts the expected perturbation. Okay, so one idea. So this type of learning is obviously cerebellum dependent, and one idea about why rehab therapy is problematic for cerebellar patients is that a lot of the techniques that are used in the clinic place a lot of importance. They sort of draw attention to, they kind of enhance vector error feedback. This is happening more and more, especially with the advancement of gamified rehabilitation techniques and use of things like interactive things like the Wii and other kind of motion capture technology. So one option, though, is that adaptation is not the only way our brain learns to change behavior. We have a suite. Motor learning and motor control depends on a suite of interacting mechanisms. And one mechanism that has been particularly interesting for me is reinforcement learning. And the reason we have some evidence to think that reinforcement learning may be less dependent on the cerebellum. So to start off, what am I talking about when I say reinforcement learning? What I'm talking about is a learning process that binds the value of a movement outcome to the sensorimotor state, or the movement that predicts it. So in this way, it's very similar to operant conditioning in its idea. One reason why we might think it might be less cerebellum dependent is that it eats different food, if you will. So the cerebellum dependent learning mechanism, we think, really responds to vector error information. A reinforcement learning system can run on scalar, or even its very simplest, binary error information. The other reason that reinforcement learning is sort of interesting, another reason, is that reinforcement learning can proceed in a model-based manner, but it can also proceed in a model-free manner. And cerebellum-dependent learning is necessarily model-based, and it may be that the cerebellum is important for all model-based learning, but it's possible that the cerebellum is less involved in mechanisms of model-free learning. So throughout my postdoc, we did some work trying to figure out how do we isolate, or how can we kind of get people to rely on reinforcement learning. And it turns out, I'm going to walk you through an example of how we kind of found we could get sort of nice dissociative behavior using a similar reaching task to what I showed you before. Now, the way that we do it is we need to remove the dominant source of vector error information. So in a reaching task with a cursor, that's going to probably be vision. And so what we can do is just have people reach, and we don't give them the direction and magnitude of their errors. Instead, we have them reach to a target, and we give them a binary signal. You either hit the target or you didn't hit the target. And what we can do is utilize methods of shaping behavior to now change the direction of the reach to get essentially the same outcome. So they start by reaching the target. Yay, we want them to change their behavior. So we stop reinforcing when they hit the target. And then we kind of push them in a direction that we want. So it's in its implementation is a little bit simple. But I want to highlight one last reason why I think reinforcement learning is an interesting idea for cerebellar ataxia. So here are the same reach trajectories I showed you before from the sort of more severely affected patient example. And the thing I'm hoping you'll notice is that the reach trajectory I've highlighted in blue is a lot, better than the reach trajectories shown in red. And this is the thing with cerebellar ataxia and movement disorders more generally, which is it's not a capacity problem, right? If you ask somebody with cerebellar ataxia to make the same reach and the movement velocity on all these reaches is constrained, some percentage of those reaches will look kind of quote unquote normal. And that to me is really fascinating, because I want to know, what is the system doing on those reaches? As a scientist, I'm very interested in that. But when you think of more clinically, idea is that I can actually be a bit agnostic about what the system's doing. I just want the system to do whatever it did on this blue reach more often. And so, what this tells me is that it's not really a capacity problem. You can kind of think of this in a very, simplistic way as kind of a statistical problem. And that's what reinforcement learning does. It biases behavior to a conditioned output. And so one thing that I wanted to know is can I just kind of can I sort of leverage this capacity, this residual capacity in the system, and just sort of condition them to produce a behavior in a stereotyped manner? So the first thing we did, of course, is we need this sort of proof of principle idea that indeed, it's that they can learn that individuals with cerebellar degeneration can learn with binary feedback. And so back in 2016, during my postdoc, the first thing we did is we had a group of patients with cerebellar degeneration perform basically the same task I showed you before. So they had to reach to a target and learn to counter a 15-degree rotation. And we did it within subjects design and had them do kind of 2 versions of this task. And really the only difference between the 2 versions is that in the version I'm calling the adaptation condition, participants could see the cursor, the rotated cursor, so they had vector error feedback. And then in the reinforcement condition, we took that away. The reinforcement signal was present in both tasks, so the target just turned green when they executed the correct movement. And so really what we're testing is the second that they have vector error, do they stop? Can they no longer learn? And our method of shaping that we used is what we called closed loop reinforcement, but basically it involved essentially taking a running average of the rewarded dimension of the task. So in this case, it was the reach angle, which you can see kind of cartoon plotted here on the y-axis over trial number on the x. And if we take a running average of the reach angle, the way that we can kind of induce, we can kind of shape them to rotate their reach is that we start reinforcing only reaches that fall within their sort of mean reach angle and the target that we want them to hit. In both the adaptation and reinforcement conditions, the intervention phase was followed by a short retention block of 100 trials where we took all feedback away and asked them to just keep reaching to the target in the way that they thought was accurate. So what we found can be seen here. So again, I have reach angle on the y-axis over binned trial number on the x. And our cerebellar group is shown here in magenta. Our group that is matched for age and biological sex is shown in black. And you can see that although there's an offset at baseline, if you look at kind of a delta value, the 2 groups actually learn about the same amount. And what's really nice is that the cerebellar group, when we take the feedback away, they don't immediately revert back to baseline behavior. They're able to sort of recall what they were supposed to be doing. And this after effect was in stark contrast to what we saw in the same group of participants, but in the case where they had a cursor. So now the same participants did not show any evidence of learning at all. So this was like a great proof of principle, but one thing that I hope you guys kind of think about, and I'm throwing shade at myself as much as everyone else when I say this. No one really cares about reach angles. So the next question that I wanted to ask is, okay, this is great, but can we use this to do something a little bit more complex? Can we use this to change something that is sort of about their behavior that is very relevant to their ataxia? And so I want to come back to these reaches that I showed you before. So as a first step, we spent some time looking at what is different about the good reaches compared to the bad reaches. We're staying in reaching for now. And one thing that you may notice is that if you constrain the start and end point of a reach, reaches that are better coordinated and that are straighter, if you will, have a shorter path length than the reaches that are curvy. And so we thought, okay, can we use the same principle idea of an intervention here, this sort of closed loop reinforcement intervention? Can we use that to now reinforce reaches with a shorter path length to see if we can get them to improve their reaching behavior? So what we did for this task is we tried to move it out of take an opportunity to sort of broach, and I use the word broach very intentionally, broach real world movement. This is still very far from that, but at least move into 3D space. And so the way that we did that is we used 3D motion capture and interfaced it with a virtual reality environment. And the nice thing about virtual reality that's good for this is that for reaching movement so far, we're operating in the space where we're removing visual feedback, and VRs, that's what it allows us to do. Now, the actual environment we had participants perform the task in was not this bleak, I promise. I just use this for illustrative purposes. But basically they had to do, was very similar in scope to the previous task. They had to make target directed reaches from a home position out to a little target bubble. They had to pop the bubble. And sometimes we gave them a cursor showing them where their index fingertip was. And then we compared that to a reinforcement case, and this allowed us to remove the cursor and provide reinforcement feedback. So our reinforcement intervention, we started with a baseline phase where they could see their hand, followed by a baseline where we take the cursor away and ask them to just reach in a straight line as best they can. We then started our intervention. And so here again, the important thing here is that we need them to solve the credit assignment problem. So we tell them, we're looking for you to reach in a specific way. My instructions are always, I'm not going to tell you what I want you to do, but I am going to tell you if you're on the right track. And so if you make a reach from the home position to the target, and in this case, our reinforcement signal was a pleasant tone, you hear the ding, that means you did great. Try to keep doing whatever you just did. If you make a reach to the target and you hear our sort of a negative or error signal, I can only really describe as like a sad noise. It's not particularly aversive, but it just kind of sounds sad. If you hear the sad noise, that means try something else. And then we followed that by with two retention blocks. So at first we just removed the reinforcement signaling, but kept the cursor removed. And then we tried a short block where we turn the cursor back on to see, are they still able to recall a little bit of a better movement, or do they immediately go back to baseline? Importantly, again, we used a within subject task design with a counterbalanced order, where we wanted to compare this intervention to something that mimics what is done in the clinic with these patients. And what is done in the clinic with these patients is a lot of practice. It's a lot of just keep trying to practice, and the idea that if you just keep practicing, maybe you'll get better. And so we followed basically the same task design, but our intervention phase was they still couldn't see their hand. But we just asked them, there was no reinforcement feedback. We just asked them, please keep trying to reach as smoothly as you can. So what did we find? Okay, so what I'm showing you here is the path length of reaches on the y-axis over binned trial number on the x. Path length is expressed as a percentage of the optimal, or what is essentially in this case a straight line path. I appreciate that no one reaches in a straight line, and I'll come back to that in a moment. But we are expressing it as a percentage because the position of the home position and the target position were based on a certain shoulder elbow angle configuration. And so depending on everyone's anthropometrics, those were slightly different positions in space. But suffice to say that 100% would have been a perfectly straight line between the start position and the end position of each reach. And what you can see is that at baseline, our group was on average about a 30%, a little bit less, greater than a straight line. But by the end of the intervention, they got down about 10% to 20% greater than a straight line. And why this is notable to me is because we had a group of matched age and sex matched controls perform a bunch of reaches in this environment. And controls tend to reach between 10 and 20% greater than a straight line. So what this told me is that I could get patients to at least on average the upper bound of what we might consider as within a healthy range of reach coordination. The retention phase, you can see, this is a very short intervention, so we weren't expecting super spectacular retention, but we do at least get some favorable results here. They are able to hold on to it. It does decay. And then one thing is when you turn the feedback so they can see their hand again. They do get a bit worse, but one thing that's nice is that they don't immediately go back to baseline. So that suggests that even in a relatively short intervention, we might be able to have, they might have learned something that they can at least kind of, that can be triggered again. So that sort of is a little bit encouraging, and we can talk more about kind of where to go from there later. But I do want to talk about our control case here. So this is our massed practice control, and what you see is a very different behavior. So again, at baseline, they're about 30% greater than a straight line. But by the end of the intervention, there was really no change in behavior. If anything, it's very highly variable. This is between subject variants, mind you. But they really didn't get any change at all. And of course, they're not retaining anything because they didn't change any behavior. So the nice thing I forgot to mention too with the reinforcement is that you see at the beginning, you see this sort of behavior. You can tell that they're exploring the space. So they are trying out different behaviors, and then when they capture the one that seems to be getting them a reinforcement, they're able to kind of exploit that feedback. Okay. So both of these studies were really nice kind of proof of principle ideas that maybe this can work. And there's a lot of directions we can go from that. But one thing that struck me is that despite the fact that on average, we tended to see learning in these populations, there was a lot of variability across individuals. Okay, so what I'm showing you here is in panel A is the total learning, so sort of where they ended up at the end of the intervention, normalized to their baseline for each subject in that first two-dimensional reaching study I showed you. And then in B is the same idea, but now for the subjects in the path length experiment I showed you before this slide. And here in a gray shaded region is where the controls, this is sort of distribution of control behavior in our 2D task. I've colored each dot based on the limb function subscore of the ICARS, which is an ataxia symptom severity score. It's a bit clunky, but I think it at least demonstrates the point that I'm trying to make here. And then each symbol represents the sort of etiology of our patients. And the thing that I'm trying to show you here is that we see variance in learning. Some people learn well. Some people do not learn well. And of course, when you see something like that, the low-hanging fruit is, okay, is it just the more severely affected individuals who aren't learning here? Is it a particular etiology of disease that can explain this? And the point here is that the low-hanging fruit doesn't explain this immediately. Granted, these are small sample sizes, but it's not immediately clear that it's always our worst person is the person who's not learning. So one thing that one of the lines of research in my lab right now is that we're trying to understand is there anything we can understand that predicts what's going on in learners versus non-learners. And so starting to think about this, back in our 2016 paper, we had developed a mechanistic model of reinforcement learning, and I'll walk you through that quickly. So basically we modeled, remember this was the task where you're trying to change the reach angle in response to closed loop reinforcement feedback. So the way that we modeled it was that on every, the reach angle that was produced on any given trial was a function of some internal estimate of whatever the person thought the correct reach angle was, plus some amount of variance. And critically, what we hypothesized was that the variance we could kind of largely bin as coming from two broad sources, remaining agnostic as to sort of what specifically each of those sources were. But essentially, the point is that one source, we called it exploration variability, but essentially it's just how much of that variability do you know about and can correct for? And then the rest of the variability is coming from essentially, we called it sort of motor noise. You could think of it as sensory motor noise. But the point is, it's variability that the system kind of can't estimate, can't correct for. And the way that you update, of course, is that you execute your reach, you get some sort of feedback, you're going to update your estimate of what the correct angle is based on whatever your previous estimate was, plus you can only really update based on variability you know about, right? Now, critically here, of course, this is sort of multiplied by our reinforcement. We are treating if you just don't update, you're not going to update if you don't get reinforced. You're only updating if you do get reinforced. There's some nuance there to think about, but this is meant to be a very simplistic kind of model. But you can already hopefully see that, you know, what the relative values of our distributions for exploration versus noise terms are going to have on behavior. So if you have a lot of your variance is coming from the distribution that the system can't estimate, you know, you're not going to learn very efficiently. So in our 2016 paper, we fit basically the standard deviations of the distributions that produced our exploration and motor noise draws on each trial, and did that for each subject to look at kind of where the parameter values most fit. And you can see that there's a bit of a group difference here. Our cerebellar patients tend to skew out on the side of having more of their variance coming from noise versus from exploration. And to give you an idea of kind of how our model predicts that should affect behavior, you can see here each square is heat mapped as the average prediction based on 10,000 simulations running the model. So it's not every simulation it's not on every trial, every run of the task you're gonna like fail, but it's on average if you have higher motor noise, you're gonna do a bit worse. And you can see that our patients are kind of skewed out in this range, where some people kind of could still do well. Some people are kind of predicted that they're really going to struggle. And so, you know, modeling's great and it's fun and I like doing it. But I think it's important to try to think about what does this actually mean for behavior. And so one thing I wanted to do is think about, can I test this behaviorally? And so thinking about, you know, what is a behavioral marker of the relative, the sort of relative motor noise to exploration noise distribution? Well, what this sort of means is that essentially, if you have really high motor noise relative to your exploration noise, it means you don't have a good sense of your movement. You don't have a good estimate of what you're doing. And if I think about kind of how we kind of quantify that during these are active voluntary reaches, thinking about maybe what's going on in our cerebellar patients, that means that they might not have great sense of their movement. I started thinking about a measure that we kind of sometimes is termed active benefit. So active benefit describes the improvement that we see in sort of somatosensory or proprioceptive acuity when people are making perceptual judgments of active or voluntary movement versus movement that is passively induced either by an experimenter or a robotic device. And some prior work from Nasser Bonpuri when he was working with Amy Bastian showed that this might be linked to cerebellum-dependent prediction. And critically, what Nasser found in his paper is that on average, people with cerebellar damage don't show active benefit. They are unable to leverage active benefit. And one thing that sort of struck me about that is I had the opportunity to collaborate with Heidi Weeks when I was working with Amy Bastian, and we tried to replicate Nasser's results. And what I'm showing you here is essentially we had people sort of localize their fingertip position, and I'm plotting the area of a confidence ellipse essentially around their judgments. And we compared passive, their localization after passive movement to their localization after active movement. Each bar here is an individual participant, or cerebellar group is in blue, matched controls are in black. And then what I'm showing you on the bottom is really what I want you to pay attention to, which is the sort of ratio of active to passive. And anything below a ratio of 0.6, we classed as showing active benefit. Now, what you can see is that most of our controls showed active benefit, although not everybody. But here's the thing with our cerebellar group, is that a lot of our cerebellar patients also showed some residual active benefit. And that in truth, we did not replicate the prior group difference. There was no difference at the group level between the degree of active benefit shown by our cerebellar patients and our controls. So that's really great, because that suggests, that might seem suboptimal, but when I see things like that, I think, sweet, I have variability I can work with. And so one question I asked is, are the people who don't show active benefit, they just their sense of where they are in space during after an active movement or during an active movement is bad enough that they can no longer map a reinforcement signal to what they did? So are the people who are bad here, are these the same kind of people that would do badly in a reinforcement learning task? And so the last study I'll walk you through is this, asking that question exactly. So we had a group of individuals with cerebellar degeneration come into the lab. We had them perform 2 tasks. The first task was reinforcement motor learning, the same task I showed you, the sort of 2-dimensional reaching. They have to learn to counter or learn to rotate their reaches by 15 degrees in a counterclockwise direction. We use the same closed loop reinforcement shaping method. And then we had them come back for another test that we called the state estimation task, but essentially we were basically kind of redoing a version of what I had the chance to collaborate with Heidi Weeks on. So in this task, basically we had people in the robot, then we show them their hand, we take it away. They would either actively move until they hit a little pillow, or they were passively moved out to a point. And then we did a 2 alternative forced choice task where we would show them a cursor again. And they just simply had to report whether they felt that the cursor was ahead of their index fingertip or behind their index fingertip. And so we tested a series of probe locations and a series of shifts. And I'll go through what we found. So first, it's always a sigh of relief when you can replicate your own results, right? So we did manage to replicate what we found in our previous work. So I'm showing you here just the group mean reach angle over trial bins. Sorry, my mouse has a little lag here. Let's see if I can do it on my laptop. Over bin, and you can see that again, very similar to our 2016 paper, we didn't see a difference in learning across groups. But always nice, again, we saw that there's variance across individuals. So again, I'm showing you each patient, and I gave their broad etiology category a symbol versus controls. Both groups here, the asterisks depict a test against 0. So they both showed significant learning. It was not significantly different from each other, but we do see a nice degree of variance where some people learned and some people didn't in our cerebellar group. Importantly, just as a sanity check, we wanted to make sure that that the kinematics were roughly similar between the 2 groups, and we saw that indeed when we look at sort of peak velocity as well as the change in peak velocity, So how much do people slow down from baseline to the end of the perturbation? We found that there was more variance, and I'll come back to that in a little bit, in our cerebellar group, but on average, the groups were not different. Nicely, we also replicated our prior work with Heidi Weeks, so we found that there was variance in the degree of active benefit that we saw. So how did we quantify active benefit? Essentially, we used, the nice thing about a two alternative force choice task is you can fit the data to a psychometric function. And what we took out of this as a degree, as a measure of their acuity in the active and passive movement conditions, was the just noticeable difference. Okay, so that's the difference in cursor shift between the 25 and 75% probabilities. So essentially what this describes is how different do the stimuli need to be for you to sort of reliably shift from saying something's ahead versus behind. Okay, and we compared, we took the difference between the active and the passive condition. Okay, and so when we look at the just raw J&D between the groups, you see that there's not significant difference between the groups. And when we look at the active benefit, again, at the group level, we don't see any significant difference. There's a lot of overlap in the distributions, and we get some variance. And so that's exciting, because when I see variance, I think I can do regressions. So we looked at, to answer our main question, we looked at the relationship between the degree of active benefit that individuals showed. One thing I should say is anything below 0, we were a little bit more lenient in what we designed as what we're taking as active benefit here. Anything below 0 is showing active benefit. Anything above 0 is, we're saying, is not active benefit. This is a little more lenient than the Heidi Weeks study. But we looked at our relationship between active benefit and RL. So we used multiple regression and looked at a number of factors. So we looked at the degree of active benefit that in our patient group that they showed on our somatosensory task. But we wanted to loop in some other potential predictors as well. So we took their ataxia severity, which I'm using the limb function subscore of the ICARS here. And then we also took, as a sort of additional measure of their severity that is perhaps a little bit more sensitive to this particular reaching task, we looked at the baseline variability in reach angles. We also looked at the change in reach velocity as a potential predictor. So one thing with ataxia, as many of you know, is that, if we think that it's a problem of predicting the dynamics of the reach, if you just slow down a lot, reaches can get a little bit easier to control. And so we wanted to make sure that it wasn't just people who tended to slow down more that were doing better. And then we also looked at age as a predictor. So our overall model was significant, which is nice. And indeed, it turned out that the only significant predictor was active benefit. So I'm showing you here, just for illustrative purposes, a very simple, the simple linear regression results. None of our other predictors within the group were significant. Now, remember that we saw a similar spread in active benefit and a similar spread in total learning in our control group. So the question here is this relationship between active benefit and RL, is that specific to individuals with cerebellar damage, or is this sort of a universal phenomenon? And seems like it is specific to cerebellar damage. So when we ran the same kind of multiple regression model in our control group, we found that none of these, obviously we didn't include clinical severity, none of the predictors were, like, the model was not significant. And that is interesting and begs some further questions because you look at some of these people here, we had some controls who did not show active benefit, and yet they learned quite well. So what this tells us maybe is that there's something about damage to the cerebellum that sort of prevents them from being able to kind of compensate in the way that maybe controls can compensate. But that's something else that we can talk about in the discussion. So I'm going to conclude here. So we show kind of three things today. The first is that we think we have evidence that many people with cerebellar degeneration can use binary reinforcement to learn to alter their movement, but a small percentage do not. And it's possible that in this population, the capacity to learn with reinforcement signaling is related in some way to the degree of active benefit, which you could stretch out as a measure of the integrity of predictive state estimation in these individuals. And I hope that some of you are thinking, hold on, wait a second, Amanda, isn't that predictive modeling or that predictive state estimation also the problem in adaptation? So is this all just the same? I will remind you, however, that some of our previous work showed that across the board, our cerebellar patients, really just the same patients who could benefit from reinforcement learning conditions did not learn when we provided vector error information. And so what this suggests to me is that, again, it's not that reinforcement, I think any motor learning probably depends on some degree we could think about predictive state estimation. This particular system maybe doesn't stress it quite as much as adaptive learning. And what this tells me is that maybe kind of potentially cuckoo idea, but maybe there's this critical range in the degree of cerebellar damage. And this is the thing when you're dealing with degenerative patients, right? You haven't shut off cerebellar output. You've made it unreliable and crummy. So maybe there's a critical range in which sensory prediction has degraded enough to preclude efficient use of adaptive learning, but it might be sufficiently spared to prevent relatively efficient reinforcement learning. So I'll end there so that we have some time for discussion. Basically, acknowledgements to the movement science group at Moss. In particular, I'm going to highlight Evan Snow and Chris White, who are two former research assistants in my laboratory who helped out with the studies I talked about today. This is a very flattering image of our campus. And with that, I will say thank you. And
Amanda, thank you so much.
Questions. Yeah.
Yeah, absolutely. That was wonderful. So please go ahead and raise your hand and I will call on you if you have a question for Amanda. Amanda, let me start with a naive question for you.
Mm-hmm.
The patients live with these disabilities throughout their adulthood often, and I wonder, why is it that just the normal reinforcement that's available from day to day isn't effective in helping them improve their behavior? And what do you think is about the feedback that you're giving them that makes it so that they can learn from it?
Yeah, so I think one thing that's really interesting and is I think a really fascinating question that I think about a lot is if we were truly an optimal system, why can't it just sort of naturally go to the other, if you think about this all as like interacting probability, like Gaussian curves, whatever, why can't it just sort of shift? And one thing that's sort of interesting is that with cerebellar patients, it seems like it does shift, right? It shifts to a very problematic direction, right? So we think that the phenotype of ataxia, what we sort of think about is particularly this low frequency tremor, matches almost exactly what you would expect of a system that's relying on time delayed feedback. And so what you see is the phenotype is actually the result of this problematic compensation. And it seems like the system wants to go into feedback control, so to speak, rather than something like this. And so one thing, and and it's a fascinating question. I'm not going to pretend that I know the answer about why that happens, but one thing that we did with a student who was working in Amy's lab for a bit is we did very similar to actually one of your papers, Reza, with Junizawa, where you compared kind of essentially vision motor rotation kind of task between full online feedback, endpoint error, and then our binary feedback. And we did that in controls and cerebellar patients. And what we found is essentially, what I'm showing you here is very simply is just the after effect that we saw with learning. And the thing I want to highlight is that our full online error feedback and then endpoint, sort of classic endpoint feedback, is exactly the same. So this possibly is that the reason is that in adults, and I think it's very interesting to think about the comparison between adult learners and adults with cerebellar damage, and then children, developing learners and children with cerebellar damage. You see these very different behaviors, right? It seems like in adults, and there's some other work by Josh Kashabak and others, I think some really nice work from Rich Ivory's group, has shown that it seems like the second you provide any kind of vector error information, the system kind of wants to use that. And so it's possible, I wonder if that's why. And one thing that we found the other thing I'll remind you is that in our studies where we provided vector error feedback, they were also getting the same reward signal, right? So like they couldn't leverage it then, right? Once they have the vector error, it's like you have to take that away, which obviously for me that makes me think of all kinds of questions about like how do we do this? Like reaching is great. I really want to start exploring this in axial control as well, which this problem becomes quite significant there. Yeah.
That's really beautiful, Amanda. Really fantastic. Thank you for sharing this unpublished work. It was really beautiful. Rich, you have your hand up.
Yeah. Hi, Amanda. Good to see you. Thanks for
Hi.
the talk. So again, yeah, so similar to Rez's question about, when you use the reinforcement learning to learn the perturbations, one thing, but it's especially interesting when you see using the reinforcement learning to enhance the natural movement, right, where they want to reach to the target, and especially because they. Once you gave that instructions, then they started doing this exploration. So they got bad for a while, but then they came back to what presumably is the natural behavior. And the question is, why, now they're basically just being trained to make what would be, I assume, the natural reach and what the people in the mass practice group are doing. And I just wonder if you did a control condition where you gave random reinforcement signals to the people in the baseline group. Because again, it wouldn't be tied to anything in their movements, but it would provide a similar level of motivation. And do you think they would show some sort of improvement, since you're basically trying to reinforce what I assume everyone's trying to do is just reach directly to the target?
Yeah, so that's a really interesting question, right? Like I'll blow the lead, which is I haven't done that yet. I really want to do that because I think that gets at this question of is this, because if they're really able to sort of develop a stereotypy, if you will, I could reinforce anything, right? Like I could also make them worse, in this case, we chose to reinforce the reaches that were, however you want to think about it. I always put it in quotation marks better. But yeah, theoretically, that would be a really good control. I totally agree. Just to see, if it was purely random reinforcement, I should expect that their behavior doesn't really change. If it's really sensitive to that feedback, we might see them developing some sort of interesting stereotypy that is unintended. Absolutely. Yeah.
But your reinforcement was based on the running average, right, of the last number of trials. It wasn't just the most recent reach that
No, so that's and that number came out of a lot of model simulations. It's of trying to find some optimal value, some optimal vector length, essentially, of like, you don't want too few trials because then it's really volatile, right? The mean is really volatile. You don't want too many. I think who's it? Maury Smith has tried to do this, and he's gone as far as like 40. Like he keeps the sort of running average of the last 40. And people never change their behavior, and it's because that at that point the mean is not responsive enough. So there's this sort of range, and I don't pretend that 10 is like the optimal value. It's just it's what we found based on simulations, but I do think that there's some optimal length there to go back. Yeah.
Well, thanks a lot.
Thanks. Yeah.
Thank you. Thank you so much, Rich. Mikola, do you have a question?
Thank you very much. Can you hear me well?
Yes.
Great. Thank you very much for a very interesting talk with the emphasis on potential application in multiple sclerosis. I was wondering, what's your take on kind of clinical implementation of the research you are doing? Because, on one hand, we have, all those scoring measures that are not very highly reproducible. And on the other hand, we have pretty much everybody having a touch screen and being able to test something, quantitatively. So what would you recommend for a tool for monitoring or measuring ataxia using touchscreen in clinical settings?
In touch screens. Yeah, that's tough. I suppose I'll give you kind of two answers of where my mind goes, and I hope that one or both of them might provide some kind of answer to your question. And if they're not, please hold me to it. So in terms of my work, I think I kind of err on the side of not of maybe underestimating how much I think it's useful in the clinic. I think right now what we've shown is that for these patients, in terms of intervention, less feedback is better. That's sort of the rule. I think that at the least, in terms of how the optimizing that less feedback, I think there's still a lot of work to go. I also, as I mentioned, we're still, a lot of this work is in reaching. A lot of the actual treatment targets for patients is axial control issues, and I think there's like some complexity to applying this. In terms of your question about tracking, yeah, I think that's really tough because you have to recognize that in the clinic, people are operating under very different set of incentives and constraints than we are in the lab, right? They have an hour. It's got to be really user friendly. It's got to be really reliable across different evaluators. So in terms of using touch screens, it's sort of tough, right? I think I think one thing that's sort of useful, I don't even know if it needs to be a touch screen, but one thing I find with clinicians here, I end up doing when I'm with them is I do a very simple thing, because there's sometimes you get these patients coming in and it says, oh, they have ataxia. And the assumption is that it's a cerebellar, it's purely cerebellar. And that's not always true, right? Ataxia can be very mixed. You talked about MS, that's a very mixed case. One easy test you can do, so in the ICARS and the SARA, there's like the finger to nose test. One thing to do that I think is useful to tell you sort of how pure cerebellar it is that you do the finger to nose with their eyes open, do the finger to nose with their eyes closed, like the patient's eyes closed. And what you'll find in at least in my experience, and maybe I'm overgeneralizing here, but the more pure cerebellar patients I see, when they do it with their eyes closed, they miss the finger. They're going to miss their finger. They're going to miss their nose. They can't see the target. But what you'll find is that the tremor, the low frequency oscillating, tends to reduce substantially, if not disappear. And that's because they don't have that signal that's triggering them to start getting stuck in this correction loop. I don't know if either of that answers your question.
That was really interesting, Amanda. Yeah, that was wonderful. Thank you for sharing that. I have one more question. Yeah, I wonder if you can talk a little bit about your really interesting finding. You've reproduced it twice now on this, which you've described as active benefit,
Mhm.
and how there doesn't seem to be a difference among the patients, and although there's variance among patients and healthy controls, but if I understood it correctly, we don't now, based on your data, we don't think this concept of active benefit is a marker of cerebellar disease. If maybe I'm overgeneralizing, can you comment on that and describe it a little bit?
Yeah, yeah. Yeah, I don't know. I think I think it's one of these markers that I think the thing when you have variance is to remember that a lot depends on the specific sample you have. So to compare, say maybe our two studies with the Bonpuri study is in the Bonpuri study, it maybe it just worked out that the sample of patients they had tended to skew on the side of not having active benefit. Nasser's paper is very beautiful. I mean, he does this sort of very beautiful manipulation where he makes the movements unpredictable in the control group and finds that he can sort of abolish on average their ability to use active benefit too. And I think that that's a really powerful and nice illustration. What I would say is that I think the picture of what the cerebellum is doing in somatosensory or proprioceptive acuity is perhaps a little bit more complex. Because one thing that is also important to remember is that we see cerebellar patients. It's not that their deficit is restricted to active movement, and that they never show passive problems. If you compare passive localization when the limb is stationary versus like you move them passively and then they stop moving and then you localize, and you compare that to active, like they have to localize while they're being passively moved, they do show like they're worse during movement, right? And we also see there's also a nice body of literature, I know Rich has done some of this, that shows that we see these sort of pure sensory deficits too, like tracking a visual stimulus, predicting the behavior of a visual stimulus. And so what that tells me is that there's something a little bit more general, that I think active benefit has the ingredient that the cerebellum is contributing, but it's not maybe the purest measure of that ingredient. Does that make sense? Yeah.
Yeah, and I think what you said about looking at real-time feedback control, and maybe in that context it would matter. I'll stop. Sasha, you have your hand up.
I do. Amanda, that was really interesting, thought-provoking. I have two questions. One, we should talk about offline, which is, but I think it's an important question in general, which is how to handle the vast differences in axial control versus appendicular control.
Yes, I have many thoughts on that.
I would love to talk to you about that because, you know,
Yeah.
we've been studying both the vestigial and the dentate. And so I have thoughts about that. So my question really is, it stems from my observation that, you know, in the motor control field, the investigators, some of them are actually very embodied and proprioceptively aware, and some of them aren't, right? And that's true in general. And it's true in the general population. So the people I've happened to work with in the clinics,
Sasha, we lost your voice.
I think we cut out a little bit. Yeah. Maybe I'll mute.
Whoops. So Sasha, we lost your voice. We can't hear you, Sasha. Oh, that's too bad.
Maybe Sasha, if you can hear me, maybe email me and we can absolutely talk offline. I'd really love to hear your thoughts on this. But I can't hear you anymore. I'm sorry.
All right. Well, everyone, thank you so much. Amanda, thank you so much.
Thanks, Reza.
It's
Good
wonderful.
to see everyone. Thanks
Bye.
all. Bye.